Great. So today I would like to talk about antimicrobial resistance. Okay. Thank you. So first I would like to introduce this model system that we used. This is human cytomegalovirus, and it is also known under the name human herpes virus. Many of you may be infected with this virus, but you are not aware of that because I will explain you later, it does not cause symptoms necessarily. This is quite big virus, and it is transmitted through bodily fluids. And as I said, it is widespread among population. So this virus in most cases causes mild non-specific symptoms or even no symptoms at all if you are immunocompetent, healthy person. The story is completely different in immunosuppressed or immunocompromised patients. These are organ transplant patients or patients with HIV or newborns. In these patients, symptoms can be quite serious, and finally, people can die from the disease. So quantification of human cytomegalovirus is needed, on one hand, to trigger the treatment of such patient, and on the other hand, to follow the effectiveness of the drug that was used for the treatment. Unfortunately, there are quite significant differences in results in clinical laboratories. There is an example in instant external quality assurance schemes. The results, which are plus minus 0.8 logarithm, are acceptable. In other words, if the target value is 1,000 copies per milliliter, valid results are between 160 copies and 6,300 copies. So you can see what is the range of the valid results. So we were interested, what are the reasons for such big differences in results? And we looked at two steps that are necessary for quantification of the virus. First step is extraction of viral DNA, of viral genome, and the other step is amplification of specific target sequences, which we can then measure and, based on that, quantify the virus. We were comparing 3 different extraction methods, and we observed already in this step that there are quite some differences between that methods, and we selected one of the methods to use it for further development of measurement procedure. We were quite lucky because we had standard. There is WHO standard available, and this was the virus that we took for the extractions. In the next step, we had again this WHO standard, but also NIST material from NIST, and we were looking at methods for quantification of different strains of human cytomegalovirus. This is based, as I mentioned, on amplification of these target sequences. Here we have much wider diversity of possible approaches. So we selected 2 different specific targets for quantification. We have done this on 3 different platforms. One is real-time or quantitative PCR. This is the approach that is also widely used in clinical laboratories. And the other approach is digital PCR, and we have used 2 different platforms. With these platforms, we were also using different key chemicals for amplifications and different approaches for counting. One approach is relative, connected with quantitative PCR. The other is absolute with digital PCR. And here we really observed big differences, especially in qPCR, so in quantitative PCR. We could observe fold differences between different key chemicals used or between different targets used. And in digital PCR, these differences were quite small compared to this in qPCR, because we could observe up to 30% differences in different chemicals in different targets. So one of these targets that was then selected for further development of measurement procedure, and we took both digital platforms for the amplification. These are examples of these platforms. What is important here is that the reaction is spread in many, many compartments, and we just have to count the positive compartments, so compartments where reaction was going on, and all the compartments that were used in this reaction. So based on these counts, we can then calculate copy number of the original sample. Another advantage of digital PCR is that because reaction is spread in so many small compartments, we can also find minority targets in these cases. But also any inhibitory substances that could be present in the sample are diluted this way, and they are less, they have less influence on the reaction. This procedure was working well, so we organized a small interlaboratory comparison where 3 different laboratories participated using 2 different digital platforms. And if you remember the results from clinical laboratories, plus minus 0.8 logarithm, here in this study, without any calibration, we obtained only up to 2-fold differences. To further see the performance of this method, we have used it in 8 different external quality assurance schemes organized by Instant, and in this we confirmed precision and reproducibility of this method. So in this phase, we were certain that we have a reliable method for quantification of cytomegalovirus. Then we move to the next step, and this is antimicrobial resistance. So patients with the infection with cytomegalovirus can be treated, and primary first drug of choice would be ganciclovir. And this is an antiviral agent that prevents replication of this virus in our body because it targets polymerase, viral polymerase. Additional step is that this drug should be monophosphorylated as the first step of activation, and this is also done by viral phosphotransferase. In patients with for example, organ transplant, treatment can be quite long-term because they have to follow the virus, and these patients have a suppressed immunosystem. So this is the opportunity for the virus to develop resistance. And actually, in this region where this phosphotransferase is located, this is region UL97. Almost all of resistant mutations are in this region, and they impair phosphorylation of this drug, and this way drug is not activated and it's not active anymore. Also, methods for monitoring of drug resistance are available, but these methods can be quite time-consuming. This is usually sequencing. This method is still very important for other mutations, for other changes in genome, which are not so frequent or which are not known. But in case of this ganciclovir resistance, there are known mutations which are found frequently in patients, and we have selected 3 of these very frequent mutations to see if the methods can be developed for these mutations and if they could be faster compared to sequencing. In case of resistant human cytomegalovirus, there are no reference materials available. So we had to prepare our own materials, and we designed synthetic DNA molecules, which are containing, on one hand, sequence where we can find resistance, and on the other hand, there is a sequence for the method that I described previously, so for the quantification of cytomegalovirus. And we use these synthetic materials then for our evaluation of these selected methods. And you can see first method is human cytomegalovirus quantification, and then we have one mutation, A594V, and then we have wild type, so the sequence without mutation of the same virus. And on the other picture, we have the other mutation, and you can see that the methods are quite different. For example, with the method M460V, we are very limited with the development of method because we are limited with the sequence, and it's not easy to improve this method. But still, we tried to evaluate these methods. We were looking for false positive and false negative rate. We were looking for threshold settings, repeatability, and intermediate precision, of course, limit of detection and quantification, robustness, sensitivity, and finally also trueness. This is an example how we were looking for false positive and false negative rate and thresholds. By setting the threshold, you can see in one picture it is 2,500, and the other is 3,000. By setting these thresholds, we wanted to exclude false positives, which you can see under the wild type. You can see two levels of negative droplets, negative partitions, but these are false positive. And then if we increase this threshold too much to avoid all the false positives, we can lose too many true positives. So this is something that we have to work on and to decide which is the most appropriate threshold to have the best results. Then we went on with repeatability and intermediate precision. For example, we did 8 dilutions of DNA in 5 replicates, and that was done on 3 different experiments on 3 different days for all 3 methods. And based on this, we could estimate the limit of detection and limit of quantification of these methods. And you can see that the methods have limit of quantification. For example, method M460V, the limit of quantification is around between 70 and 80 copies per reaction, and limit of detection is at 20 copies. If you look at the method A594V, then you can see that the limit of quantification and limit of detection are the same, around 20 copies. We also looked at the linearity of methods, and it was okay. But then we also looked at the sensitivity, and with these results, we were not too satisfied because we had quite a big bias to the quantification of CMV method. Nevertheless, we got the opportunity to participate in external quality assurance scheme. So we got samples with the resistant virus in this, so samples with resistant viruses, and we were able, so using previously developed method of quantification of cytomegalovirus and these new methods for resistance, we were able to correctly identify samples with the mutations, 2 of them. Again, there is no calibration involved in these measurements. So to conclude, using these 3 mutations that are giving resistance to ganciclovir in human cytomegalovirus, we have explored how measurements of these specific known sequences could improve measurements of infectious agents, including their resistance. And we believe that this way diagnostics could be improved and be more comparable between different clinical laboratories. Digital PCR is a method that has the potential to be used as a reliable method for diagnostics, but also as a reference measurement procedure and as a method for value assignment of control or reference materials and for materials used in quality assurance schemes. And this is for different areas, not just infectious agents. The same method can be used, for example, for biomarkers of cancer, for other genetic disorders, but also other areas like in plant pathogens or in environmental samples. Before I finish, I have to say thanks to our funders. This work has been funded through international projects, but also by some national support. And if you just have money but no people behind, it is not okay. So what I presented are results of 2 big projects. And here I'm just highlighting people who were more involved in this cytomegalovirus work, because we have worked, or other partners worked on other organisms. And maybe I should just mention Jim Haggett, who was the coordinator of the projects, and the same thanks to everybody else. And thank you for your attention.